Good day, this is Professor Will, CBMD, PhD. Today is Tuesday, March 26, 2019. It is 21:41 seconds Eastern Standard Time, and this is my reading on a brief description on biofilm. So let's get started here. Biofilm. Biofilm is a mixture of microorganisms living as a community attached to a surface. This three-dimensional layer is a diverse microorganisms tend to form on surfaces immersed in a liquid, usually a flowing liquid. Biofilms form on virtually any surface, but they are most commonly in the following places: water distribution pipes, cooling water distribution lines, toilets, drains, showers, manufacturing equipment, oil drilling equipment, ship and boat hulls, and the rock, sediment, and plants, and streams. In medicine, biofilms raise health concerns by growing on prosthetic devices, contact lenses, teeth, catheters, and feeding tubes. In nature, biofilms are very resistant to harsh conditions. They have been found growing in the acidic steam pools in Yellowstone National Park as well as the glaciers in Antarctica. Biofilm is a good example of symbiosis, wherein organisms live in close association, either in a beneficial relationship or in a relationship in which one member is harmed. The symbiotic relationship found in biofilm benefits all or most of the microorganisms living in the layer. The film layer protects the microorganisms within it so that they can withstand conditions they might not tolerate outside the biofilm. Biofilm almost always contains a heterogeneous mixture of any or all of the following types of microorganisms: gram-negative bacteria, gram-positive bacteria, archaeans, fungi, algae, and protozoa. Many non-living things such as dirt, debris, organic and inorganic compounds, and dead microbial cells also become trapped in thick biofilm. The cell structure of biofilm. Cells in biofilm communities have 5 main advantages not available to planktonic cells, meaning cells that float free in liquids. First, a mixture of biofilm cells and their secretions provide each other protection from strong chemicals and high temperatures. Second, the biofilm layer that adheres to a surface enables a community to stay in place in fast-flowing liquids while planktonic cells wash away. Third, biofilm structure provides a food storage mechanism. Fourth, biofilm efficiency pulls nutrients out of nutrient-scarce liquids such as drinking water and makes them available to the community. Fifth, a biofilm community forms a cooperative relationship among its members. In this mutualistic relationship, members share food sources, growth factors, enzyme activities, and protective secretions. Biofilms have been described as mini ecosystems because of the interrelationships among biofilm species. As microscopy has become more sophisticated in showing the minute details of the microbial world, the complex of biofilm has been revealed. We usually find that well-fed biofilms are unstructured and flat, while less favored biofilms are highly structured, and most importantly, biofilms in several natural environments are seen to be composed of tower and mushroom-shaped microcolonies interspersed between open water channels. These observations led to more detailed studies on how biofilms from a seemingly independent organism composed of many different types of microorganisms. Biofilm formation starts with the adhesion of organic and inorganic compounds to a surface. Either these compounds diffuse from the liquid or the liquid's turbulence forces them onto the surface. The accumulation of compounds at the solid-liquid interface is called a conditioning film. A conditioning film provides a higher concentration of nutrients for microorganisms to use as they attach and begin to build up. In the second step, a few planktonic cells flowing past the conditioning film stick to it. They begin producing a sticky substance which allows them to remain stationary even though water flows over them. Sometimes the water flow is quite vigorous, but biofilms are known to withstand being washed away even in fast-flowing and agitated waters. This attachment and conversion of a swimming cell to an attached cell takes just a few minutes but occurs in 2 stages. In the reversible attachment step, cells form a weak chemical attraction to the surface. Cells may bump into the surface in this step by chance or by their normal Brownian movement, the random movement of bacteria and water. Then the van der Waals forces maintain a weak attraction between the cells and the surface. Rapid flows that produce high shear easily dislodge cells in this reversible attachment step. A small number of cells do not rinse away. However, these persistent cells initiate the third step, irreversible attachment, wherein a permanent layer of the cells biologically bind to the surface. Once the cells attach to the surface, they are no longer planktonic. They are called sessile cells. The word sessile is from the Latin for to sit. Additional microorganisms attach to the surface to form a monolayer of cells. This monolayer represents the 4th step in biofilm formation. Other cells soon attach and the film thickens. The thickening of biofilm is the 5th step in its formation. In the final and ongoing step in biofilm formation, the cell numbers increase and certain bacteria within the colony excrete a gooey exopolymer, a long-chain molecule excreted by a cell called extracellular polymeric substances, EPS. Early research in biofilms referred to the EPS as the glycocalyx, Glycocalyx is now known to be a more specific polysaccharide capsule made by some but not all bacteria. EPS contains a complex mixture of large polysaccharide sugar chains with a smaller portion, about 15% of protein. Pseudomonas aeruginosa is one of the main producers of EPS, but other species contribute. Only attached cells produce EPS, as if they sense that they belong to a stable community, while free-floating cells do not produce EPS. Microbiologists have shown that EPS also helps the first layer of cells attach to an uncolonized surface. A mature biofilm is a complex and heterogeneous mixture of alive and dead cells, EPS nutrients and other compounds pulled from the water, inert substances, and material corroded from the underlying surface. Very thick biofilms contain layers in which microenvironments differ from the outer reaches of the film down to the attachment surface. For example, oxygen concentrations change in the deeper regions of thick biofilms so that aerobic species live near the outer surface near the flowing liquid, and anaerobic species inhabit deeper layers near the attachment surface. Today, this is Professor Will, CBMD, PhD, case Tuesday, March 26, 2019, and this concludes my brief description on biofilms. Hope you enjoyed listening. Have a great day.